Thank you very much for inviting me to present at this seminar series. It's an honor. I've been following for the last seasons, and it's great to now be able to share my work with you here. This is work that I've been done in the last 2 years, and it's based on developing models that are constrained by the connectome. So let me start by defining what I mean by connectome. Whenever I talk about a connectomics dataset or connectome dataset, I refer to the experimental measurement of the synaptic wiring between pairs of neurons across a large brain region. The connectome of a small animal, the C. elegans, a worm, has been known for a few decades now. It had been manually traced neuron by neuron because there are only 300 neurons. So it was a long manual effort, but it was possible to do it back then. But it's only been recently in the last decade that the technology has allowed us to measure the connectome of larger insects, such as Drosophila, for example, the Drosophila larva that has about 10,000 neurons, or the Drosophila adult brain that has in the order of 100,000 neurons. There are current efforts to build connectomes of larger and larger animals, so I think it's a good moment now for theoretical neuroscientists to think about what can we say, or what information can we extract from the connectome to say something about the neural system. In particular, this talk will be based on theoretical results and simulations, but the types of data sets I have in mind are those of the Drosophila. There are two big data sets that are publicly available, one from Janelia and the other one from the Flywire consortium, that they're slightly different, but they both include many neurons in the order of 100,000. And there's something important to say about these data sets. In general, when people talk about connectome, they just talk about whether two neurons are synaptically wired to each other or not. So it's a binary information in principle. Is neuron A connected synaptically with neuron B? But in the Drosophila brain, we can actually have a better granularity of connectivity. First, in this recent study shows that it is possible to predict neurotransmitter of every synapse in the network. So not only we know whether two neurons talk to each other, we also know what is the sign of that contact, whether it's excitatory or inhibitory, or at least we can have a good prediction of what that sign should be. And the second important piece of information available in Drosophila connectomes is that we also have, or we can have a good estimate of what the synaptic strength between two neurons is. Because in insect brains, two neurons often connect with each other through multiple synapses, sometimes in the order of 100 synapses between the same two neurons. And it has been found with measurements that the synaptic count, the number of synapses between those two neurons, can be used as a good proxy for the synaptic strength. What happens, how strong is the effect on the postsynaptic neuron when you excite the presynaptic neuron? So that being said, what I have been doing together with my postdoc supervisor, Ashok Litwin-Kumar, is to think of how much is the connectome constraining neural activity, and how can we predict neural activity? And I've told you what we know about the connectome and what we can infer, but there are also many things that go on in the brain that we don't know. They're basically invisible to the electron microscopy scans used for the connectome. One of such ingredients is neuromodulation. We don't have any access to the neuromodulatory system of the brain based on the connectome. And the second ingredient that I'll focus mostly in this talk is the fact that we also don't know what the single neuron properties of individual neurons are. What are their physiological properties, for example, their excitability? And this is something that can completely change neural activity. So the question I will ask is, given that we have this big uncertainty on single neuron physiological properties, but we still have a good estimate of the connectome, what can we say about neural activity? Actually, relating connectome with activity has been done in previous years, and I think there are many studies, and they can be grouped into two different lines of research. One series of studies, what they do is they take the connectome and they use that as some ground truth structural connectivity. And what they do with that system is to record from many neurons, whole brain imaging, let's say of the Drosophila or C. elegans, for example, and then they build a functional map, something like the functional connectivity based on the activity that has been recorded. And the goal then is you have a structural map of how neurons interact with each other. You have a functional map of how neurons act with each other, and you can compare those two. And what has been found, just summarizing very briefly, is that there's no one-to-one mapping between the function and the structure in a neural system. And that is actually something that we expect from theory. So there's work by Lafitte and colleagues that shows that in general, predicting connectivity from neural activity is a hard problem, generally an ill-posed problem if you consider nonlinear networks. So we don't expect ever to find a perfect correspondence between function and structure. But this type of experimental studies, they can give bounds to tell us how much we expect those two to match or not in different animals. Another approach to use the connectome is, instead of using it as some ground truth for comparison, is to use the connectome in some generative way to constrain a network model and then fine-tune that network model to reproduce or predict neural activity. And this has been done in recent years. You can think of, for example, training the connectivity of a network and using some constraints based on features of the connectome to initialize the network. And there have been multiple successes. It seems to be, in some cases, more powerful to initialize models that way. But there is no guaranteed principle that using connectomics features will help for any reason. So based on these two type of approaches, I built with Ashok some theory to explore when do we expect this type of connectome constraints to work or not. So the main question we're asking in this work is, what is the space of solutions when you train or you fit a neural network model with connectome constraints, given that we don't know the single neuron properties in the system? And there's the second question that will come up towards the end of the talk, which is, if we know the connectome and we're always recording some neural activity in the system, can we say which neuron should be recorded preferentially? So the outline of my talk goes as follows. I will first present the theoretical setup to study this question, the teacher-student setup. Then I will show some numerical experiments based on that teacher-student setup. And for the second part of my talk, I will explain some theory we developed to explain why we obtain those results in numerical experiments. And finally, building on the theory, I will show the application that we can actually select the neurons that are more informative to record from. So let me start with explaining the teacher-student setup. I'm going to have two networks for the rest of the talk. One network is the teacher network, and the other one is the student network. Let's start with the teacher. This is a network that represents the ground truth. I'm using this as a proxy for a network from which we have the connectome, and we know how neurons are connected to each other. I'll assume this is our ground truth network, and I'll assume that it follows this type of dynamic. So each neuron is a rate unit, and the input current of the ith neuron is the dynamics of that input current is given by this differential equation. So there's a leaky term. There's also the current that comes through the connectivity of the firing rates of all the other neurons in the network, and we can have additional external inputs if we want. There are two types of parameters that determine the teacher network. One is the connectivity, and I'm assuming that connectivity is known and is based on a connectome that is available. And there are also a second type of parameters, which are the single neuron parameters. So each neuron will generally transform non-linearly input currents into firing rates. And I will assume the single neuron parameters are just different parameters of this transformation. So let's say if neurons are implementing a threshold linear function, we can say that each neuron, for example, will have a different bias, so a different activation threshold, and also a different gain. something, but I will play with different transfer functions in the rest of the talk. So this is our teacher, our ground truth. We won't be touching the teacher once it's set. And I'll be playing with the second network, which is the student network. So I'm building a surrogate recurrent network, and I'm building it to have the same type of dynamics with the same connectivity, but the single neuron parameters, this set of theta parameters, gains and biases, are different in principle. So the goal, now we have these two networks. What I'll do next is to measure something about the activity of the teacher. So let's say, for example, I record the activity of these 3 neurons in the teacher. Now I can go to the student and I can train all the single neuron parameters in the student to make the activity of those same 3 neurons in the student equal to those in the teacher. So that is the goal, and that's what I will generally do. I will use standard training techniques based on gradient methods for training the single neuron properties, gains and biases in this example. And I can tell you already that training always works. I can always make the student reproduce the same activity as recorded in the teacher. But the question is, once I've trained, what happens with the neurons whose activity I didn't record? So from the remaining 5 neurons that are not available to us experimentally, are we able to predict in the student the same activity or not? That's question number 1. And question number 2 is, if that activity is the same, so if we're able to predict activity of neurons we didn't record from, are the single neuron parameters of the student equal to the single neuron parameters of the teacher or not? So those are the types of questions I'll be asking and the setup I'll be playing with today in this talk. So let me show you one numerical example. We wanted to use something that's relatively biologically plausible. So we're using here as a teacher in this first example, and a recurrent neural network that's able to solve complex spatial temporal tasks similar to those used in neuroscience studies with animals. So this the task doesn't really matter, but I want to show you the flavor of what this teacher network is doing. So we have a network of 300 units, and I'm showing you here the readout. The readout is a linear combination of the firing rate of all neurons in the network. And if there's nothing going on, the readout stays at 0. But then at any random point in time, the network can receive an input pulse, and that input pulse can come along pattern 1 or pattern 2. And if the input comes through pattern 1, the readout of the network produces 2 cycles. And if the input comes along pattern 2, the readout of the network produces 5 cycles instead, and then goes back to 0. So this is similar to a cycling task used on monkeys in the Mark Schussen lab. So that's the task. Let me now tell you about the structure of the teacher recurrent neural network. So I'm using as nonlinear functions of single units, soft plus. It's just a smooth threshold linear, a smooth ReLU rectification of the input towards the output. And again, I'll use 2 parameters to define single neuron activations. One is the bias, and the other one is the gain. And then about the connectivity, this is the thing we know about the teacher, the thing the student has access to. We will be using connectivity with excitatory and inhibitory units that follow Dale's law. And it's also relatively sparse. It's about 50% sparse. So this is our teacher. I'm assuming this is the network from which we have a connectome available. Now let me build the student. So the student will have exactly the same connectivity matrix, but the gains and the biases of single neurons will be shuffled across the population. So let's start then by training these gains and biases of the student to reproduce something of the teacher. The first question is, this teacher is actually solving a task. It's producing a motor output. So what if we train the student to produce that same motor output using the same readout? That's what I did here. So this is the loss over training epochs. Each colored line here corresponds to a different initial shuffle of the gains and the biases. And the loss is defined here as the error between teacher and student in the readout. And you can see that as a function of training epochs, no matter how we shuffle the initial parameters, the error goes down to a very low value. I'm showing in the inset here the readout for one trial when the teacher network is producing 2 cycles. And you can see that all the student readouts lie right behind that black line. So basically all the students we trained are solving exactly the same task. They're producing the same output. The question then is, what happens with neural activity? Are we finding students that have exactly the same activity as the teacher or not? This is what I'm showing here over training epochs. You can see that we start with an initial high error in the activity of neurons, and over training that the error gets even worse and it saturates at a very high value. I'm showing here an example of one single neuron in one trial of the teacher, that is the black line. And you can see that every time we train the network, that same neuron in the student will be doing something completely different. So the conclusion of this numerical study is that even with knowledge of the connectome, the task readout is not sufficient to predict neural activity from neurons we didn't record from. Okay, but you may claim that this was too hard. We're just asking the student, we're training the student based on a one-dimensional readout of the activity. And there are 300 neurons, so maybe we don't expect to predict all the neurons based only on this one-dimensional readout. So we changed the way in which we trained the student. Instead of looking at the readout, what I will now do is to select a given number of neurons in the teacher and make the student reproduce that activity. So the first thing I'm showing here is after training, what is the error in unrecorded neurons when I recorded from one neuron? So here I'm plotting the average mean squared error in the activity of the 299 neurons I didn't record from. And this is just the summary statistics after learning. So you can see that after learning, we didn't predict well the activity of the 299 remaining neurons. We can now increase the number of neurons we record from. Let's go up to 20, and the error in unrecorded neurons stays constant at a high value. And you can see here an example of the mismatch between student and teacher. So the color map indicates the error in the activity as a function of time. And you can see that for these first 20 neurons, the error is 0. Those are the neurons I'm using for training. So the student is able to perfectly reproduce the activity of those 20 neurons, but there are strong errors in the 280 remaining neurons. Now, as we go forward and we record from more and more neurons, we found a very sharp decrease in the error of unrecorded neurons. If instead of recording from 20, we record from 60 neurons and we perform the same experiment, now, again, we're able to train. There's no error in matching the activity of those 60 neurons, but we can predict very well the activity of the remaining neurons. We can start predicting what's going on in this neural system. So we thought that's very, very interesting that there is some threshold of how many neurons we need to record from, and then we can start predicting what's going on with a neural function. The question then is, what happens with single neuron parameters? So we've seen that if we train with enough neurons, we can predict very well. We can produce students that basically do the same thing as the teacher. And we were wondering what happens with the gains and the biases. So I here separated gains and biases from the neurons we're recording and the neurons we're not recording, because as a reminder, I'm training all single neuron parameters. And this is shown as a function of epochs. And you can see that towards the end of training, even for those networks that produce the same activity as the teacher, the yellow lines here, the error in gains and biases is very high, both in neurons we're recording and neurons we're not recording. So this was puzzling to us at the beginning because we have basically teacher and students that do the same thing, but have different single neuron parameters. Finally, the last thing I wanted to do with this example is to compare with the case where we don't have access to the connectome. So what I'm showing here is the same plot as in the previous slide, the fact that after recording from enough neurons, we can start predicting well activity. I want to highlight the fact that when we train single neuron parameters, we're training order n unknown parameters. So if we have gains and biases, we're training 2n values, where n is the number of neurons in the network. Now, I'm gonna, just as a comparison, I'm gonna build a student that doesn't have the connectivity information. So in that case, we will have n squared unknown values. And just for comparison, I'm assuming we know the gains and the biases in this example. When we train the synaptic connectivity, we'll have much more parameters to train on. There's this different scaling of the unknowns. And we find that in this case, it doesn't matter how many neurons we're recording. When we try to match activity between teacher and student, we're not able to predict well what's going on. So there are some issues when you do this mapping between teacher and student when you train the synaptic connectivity, because there's no clear mapping of which neuron in the teacher corresponds to which neuron in the student. But even when we try to find the best match, we're not able to predict what's going on with the activity of those unrecorded neurons. So the conclusion from this experiment is that given knowledge of the connectivity and recordings from a subset of neurons, we can predict the activity of the unrecorded activity in the network. So the big question now is, okay, I've shown that we can use the connectome for prediction, but how many neurons do we actually need to record from? In this case, it seemed to be like 40 out of 300. But can we determine what does this number depend on? For that, I built a separate set of teachers where I'm going to parametrically change the number of neurons in the teacher network. That way we can see, as we have larger and larger networks, do we need to record from more and more neurons or not? And I built these teacher networks in a particular way, such that the dimensionality of the dynamics they generate does not depend on the total number of neurons. So these networks have rank 2 connectivity. That's the trick, to make the activity have constant dimensionality. So they're built in such a way that all these networks, from networks with 100 neurons and networks with 1,000 neurons, they are producing the same limit cycle in state space. So that when you do PCA and you take the first 2 dimensions, you basically see this clear oscillation in state space. But behind this, there are networks that are small and networks that are large. So now I'm going to run the exact same experiment. I'm going to build student networks that have the same connectivity as the teacher for all these network sizes. And I'm going to check how many neurons I need to record from, and how does this depend on the number of neurons. And this is what I'm showing here. The first thing, this is the error in unrecorded activity when I'm recording from a subset of neurons, starting with 1, 2, 10, etc. And the first thing you can see is that all lines are on top of each other. That means that there's no dependence whatsoever on the number of neurons. It doesn't matter if the network has 100 neurons or 1,000 neurons. It seems that the decay of this curve is the same in all cases. The second thing we can say about this result is that it seems that once we record from between 2 and 4 neurons, we're basically saturating. We only need to record from, let's say, 2 neurons to predict what's going on with all the other neurons in the network, independently if all those other neurons are 98 or 998. So from this type of experiments, we can conclude that the number of neurons we need to record from depends on the dimensionality of the dynamics and not really the network size. And I think this is very encouraging for connectomics research, because as connectome data sets are becoming available from larger and larger animals, we're saying that the number of neurons you need to record from to say something meaningful about what goes on in the system depends on whatever the system is doing and not how many neurons you have in total. If instead we found that it's a fraction of the network size that you need to record from, it would be a problem for mammalian systems, for example, where 1% of all the neurons in the brain, it's still a huge number of neurons to record from. Okay, so yeah, this was the conclusion, that it's the dimensionality of the dynamics that's key for determining the number of of neurons we need to record from. So I'm going to move now to theory. I've shown you these results. They're all based on simulations in my computer, basically. And now we wanted to understand better what's going on by developing some simple theory. I won't go into all the details, but the key idea is that we can always find a mapping between the activity of different neurons I in the network as a function of time and the single neuron parameters. The problem is that in general, this mapping is a nonlinear mapping, quite complicated. But one thing that we do know is that that mapping will depend strongly on the connectome, on the connectivity we have available. And there are some cases, some very specific cases where we actually know exactly what that mapping is. I will start with that simplified case. I'll show the theory for that case. And in the preprint, we then show that if we relax some of the assumptions, we still find the same types of conclusions that the theory shows. So the type of networks I'll be talking about for the theory are linear networks whose dynamics are just given by this linear system with the connectivity. And the single neuron parameters of this network are the biases. So this is just a simple linear dynamical system. We understand how it works. If you change the biases, you'll change the type of dynamics this network produces. Secondly, but that's not very important for the theory, I'll focus on the fixed point of the system, although we could also have the activity as a function of time. When you give an input, nothing changes in the theory. But just for simplicity for this talk, I will focus on, when I talk about activity, I will talk about the fixed point of this linear system. And we can find a closed form expression for the fixed point. And this is just some function that depends on the biases, and it's a linear function. So we have a mapping. The function I was talking about before for this simple system is just a matrix that multiplies the biases and gives us the value of each neuron at the fixed point. The particular mapping is a function of the connectome, and that is important. So here the dagger corresponds to the pseudo inverse. I is the identity matrix, and J is the connectivity matrix. The important thing is that we just have one simple matrix that relates single neuron parameters with activity. So that's the first step, finding that mapping. Once we have that mapping, we can actually say or predict how the learning dynamics of the biases are going to look like. So once we train the student, we're going to apply gradient descent. So in general, we start with building a loss function. Whenever you see an asterisk, that corresponds to the fixed point of the teacher. And when there's no asterisk, I'm talking about the fixed point or the properties of the student network. The loss function written in vector form is just the average error in the mismatch between the activity in the student and the teacher, just squared. And what we're doing when we train the student is we start with some random guess of what the biases are, and then we're applying gradient descent in parameter space. And this gradient descent dynamics for slow learning rates will go in the negative direction of the gradient of the loss. So this is just the simple equation. We can introduce this linear mapping between X and the biases and solve the gradient descent dynamics. And this is how the gradient descent dynamics looks like. Again, it only depends on this matrix A mapping, actually on the A transpose A. So you can see that this is A transpose A is a positive definite matrix, so that it will only have positive eigenvalues with orthogonal eigenvectors. And if we train for long enough, in the end, the single neuron parameters B should, in the student, should converge to those parameters in the teacher if this A matrix is full rank. The problem is when that A matrix is not full rank, there will be some dimensions, some parameter modes that won't be learned between student and teacher. So actually everything that determines the dynamics, like the only parameters that determine the dynamics are the singular values of this mapping matrix A. And if we have a very high singular value, that will correspond to a parameter mode along which the biases will be very quickly learned. And using the terminology of Yves Marder, we will refer to those modes as stiff modes. And then if the singular value is very low or even 0, that would be a very sloppy mode. That implies that that parameter mode will take a very long time to learn. And if it's 0 in practice, it means it will never be learned. So those corresponds to sloppy modes. Those are single neuron parameter modes that do not change anything in the loss function. So this is the simple setup we have. It's all just a linear system for this type of network. The only thing I didn't talk about in order to relate this with the previous experiments is what happens when we record only from a subset of neurons and not from all the neurons. So what if the loss is not the mean squared error of all the neurons as shown in here, but just the subset of neurons? Sorry. So yeah, let me show that. I'll go through the different panels in a second. But the idea is that if we're only recording from a subset of neurons, instead of using the full mapping matrix A, we will just be selecting some rows corresponding to the neurons we're recording from. So we're basically reducing the rank of this matrix here. So that means we're introducing fake sloppy dimensions that if we record from enough neurons, those sloppy dimensions should disappear, but because we don't have access to the full system, we are producing sloppy dimensions. So let me show you let me explain what I'm showing here. Now, I wanted to build now a specific teacher network that follows this linear dynamics. So I'm using a network with random connectivity. It's just normal a Gaussian random matrix. The only thing I'm doing is that I'm setting the rank of this matrix to be 60. So it's random connectivity all to all, but the rank of the connectivity is 60. So I can first calculate what this mapping matrix is, and I can look at the singular values of the matrix. And as we expect, when the connectivity matrix has fixed rank, the mapping will have exactly the same rank, meaning that singular values beyond 60 will correspond to 0 modes. There will be infinitely sloppy modes. And then we will have different parameter modes that will decrease to 0 slowly. So those are the modes along which the biases are learned. And what I'm showing down here is what happens when we subsample. So instead of taking the singular value decomposition of the full A matrix, I'm taking the singular value decomposition of this subsampled matrix, this lower rank matrix. The different colors corresponds to different number of recorded neurons, different M. And I'm showing here in the inset what is the angle between the subspace spanned by this low rank approximation and by the full matrix A. And you can see that if we don't record from enough neurons, these subspaces will largely be orthogonal. And as we record from more and more neurons, the subspaces become more parallel. And the key point here is that if we record on average from more than 60 neurons, we should be able to sample to well approximate all the stiff modes of the full mapping matrix. So this is the flavor of the theory, and it recapitulates most of the results we found for those nonlinear networks, although we made that big assumption that here everything is linear and we only care about biases. So the summary from the theory, I hope it was more or less clear, is that first there is a single minimum. That's something that doesn't happen when we don't know the connectivity. The biases tend to go towards that single minimum. However, there are some dimensions that will be very sloppy and some dimensions that are stiff. So in practice, even if trajectories all tend to converge to the minimum, there will be some parameter modes that are never learned. Finally, this also tells us that the required number of neurons that should be recorded depends on the rank of that mapping matrix. And that rank is basically related to the rank of the connectivity that can be linked to the dimensionality of the activity that network can produce. So now we can actually run for these linear networks, we can predict what is the error in unrecorded neurons, the error after training for a long time in recorded neurons, and what is the error in single neuron parameters. And just as a summary figure, I'm showing them all here. And you can see that if we record from more than 60 neurons for this rank 60 connectivity, we expect to find zero error between teacher and student, and we expect to have a mismatch when we record from less than 60 neurons. The other thing that we can simply reproduce with this model is the fact that even when we're predicting perfectly the activity in unrecorded neurons, we can find that the single neuron parameters between teacher and student are different. So because there are this many sloppy dimensions in parameter space, we will never be able to fully recover the exact values of the single neuron parameters. But probably we don't care. I think that's also one of the messages of this talk, is that maybe this exact value of those single neuron parameters doesn't matter. What we want is to find just the stiff modes and learn those stiff modes in order to predict the correct activity of the neural system. So with this, I would like to talk about the application. It's still in linear networks, and I will mention in a second what happens when we go to nonlinear networks. So far, I've been picking networks to record from in the teacher randomly. If I had 300 neurons and I said I was recording from 3, I was choosing those 3 randomly. But there are many ways to choose 3 neurons in a network with 300 neurons. And the question I'm asking is whether there is a better way of choosing that's not random, and whether we can predict that based on the theory. So this is just a graphic illustration of what I said before, that the parameters are related to the activity via this mapping matrix. And when we subsample neurons, when we record only from a subset of neurons, what we're effectively doing is, instead of using this full matrix, we're just selecting, if we're picking neuron I, we're just selecting the row ith, the ith row of this matrix. So what this tells us is that it's very simple to show mathematically that if you can only pick one row, you should pick the row here that aligns the most with the top singular vector of this matrix A. So this gives us a way of saying which is the first neuron that should be recorded. And then we can apply this algorithm recursively and find which is the second best neuron to record from, given that we've recorded from the first one, etc. And we think this can have applications for experimentalists, because given a connectome matrix, giving a connectome, we can calculate this mapping, and then we can diagonalize the mapping, and we can see which are the neurons that give us most of the information. So this can be done for the linear network I was talking about in the previous slide. And we can see that the number of neurons we're recording from, in order to obtain zero error in the unrecorded activity, that's not going to change by picking a better selection of neurons in general. But what we can do is optimize the error in unrecorded neurons when we select only 2 neurons. And I'm showing here in red then what that best strategy is for this linear network. Actually, we can also calculate what the worst strategy is if we want to. We can pick the neuron that worst approximates that full matrix A, and that is the blue line here. And then we can also randomly pick neurons, and that should be more or less between the best possible strategy and the worst possible strategy. But again, this is all for linear networks. In the preprint, we're actually showing that many of the conclusion, the overall picture from this linear mapping holds for some nonlinear cases. And also the numerical results we obtained are consistent with this simple loss function with one single minimum. So we can actually extend this approach to nonlinear networks. The only thing is that now we won't be able to find the exact linear mapping between parameters and activity, because there is no such a linear mapping. What we can do is to approximate. The problem is that in general, the linearization will depend on the parameters of the teacher, but we can just use some prior for the gains and the biases in the teacher. And then given that we know the connectome, we can calculate this approximate mapping matrix A. This is what I did for this example here. This corresponds to the network, the teacher network solving that cycling task from the beginning. And we can then determine in this network which are the neurons that are the most informative and the least informative, and compare with the performance with random neurons. And we can see that, at least numerically, we're able to reduce by 50% or even more for some recorded number of units when we pick from the best strategy. And actually what happens if I try to record from the worst, from the least informative neurons, is that when I try to train this network, applying gradient descent on the single neuron parameters leads to unstable solutions when we're only recording from a few neurons. So it doesn't work to record from those neurons that are not telling us anything about the function. So with this, I would like to conclude and go through the conclusion. So the first thing we found in numerical simulations is that the connectome is not sufficient to constrain the recurrent dynamics of neural networks given a dynamic task. However, we can delete this degeneracy when we record from enough neurons in the teacher network, and then predict what is happening with the neurons we're not recording from. We find that based on the theory, we can say that there is a single minimum in the loss function, and what matters are the stiff and the sloppy modes along that single minimum. If you want to think about it with other terms, what I'm saying is that it's the Hessian around the minimum that determines everything about the dynamics. And we can use that Hessian to prioritize which neurons should be recorded in neural systems from which we have a connectome. I would like to thank my postdoc supervisor, Ashok Litwan Kumar, and other lab members. And if you're interested in knowing more about this work, feel free to check the preprint. Thank you.